Hello and welcome everyone. In this podcast we're going to talk about dosage compensation of the X chromosome. Remember, in previous sessions we talked about the fact that if you look at humans, a female has 2 X chromosomes and a male has 1 X chromosome. Yet, what they express from each of these chromosomes is the same. So we get the same amount of X chromosome expression. Yet we have twice the amount of genes in females. And we know if we were to have twice the amount of X chromosome expression in humans, that would be a lethal situation. And so that's something we of course would want to avoid. Previously we talked about how humans accomplish this feat of only having one amount of X chromosome expressed. And this is true for most all placental mammals. And the way that this is accomplished is their 1 X chromosome is inactivated. We're going to come back to this and we're going to spend a fair amount of time talking about this in just a moment. Second, and we haven't talked about this one yet, and this is how C. elegans accomplish this. And they do this by their XX individuals, the XX females, only express at a half rate. So for C. elegans, the male is going to express the X chromosome at the, say, well, let's call it the average rate. And the female, the XX, will express each of the X chromosomes but at half the rate, half the speed. Drosophila melanogaster take another unique way of accomplishing dosage compensation. And for them, the male, XY, the XY male, expresses their X chromosome at twice rate. So the female XX they don't mess with, but in this case the male takes their X chromosome and expresses it at twice the rate. Thereby both females and males in fruit flies express the same amount of X chromosome genes. We're not going to have time to go into these number 2 and number 3 any more than what I just told you, so make sure you know this much here. But I want to spend a little bit more time talking about placental mammals. So X chromosome inactivation. We've talked about this before, so I want you to go back and review that material from the epigenetics sessions. As a brief background, but I need you to kind of expound on this a little bit more. Remember that an embryo starts off as XX, expressing both of those chromosomes. Early in embryonic development, one is inactivated and the other remains active. And then these each will continue to grow with the same one inactivated. And that's how we get traits like the calico cat. And we talked about that sweat gland disease. So go back and review some of that information. In about 1949, researchers, one of them by the last name of Barr, discovered a densely stained body inside of the nucleus. And they only found it in female cells. Now at that time they didn't know what it was, but later on, about 10 years later, 10-15 years later, somebody else came along. Then about 10-12 years later, a few other scientists started looking at these, and they proposed that this densely structured body was actually the X chromosome. And the reason you could see it is because it was inactivated. And so they termed these structures Barr bodies. So I'm going to draw a couple cell nuclei here. No more than a couple. I think that should be enough. Let's look at what the nuclei would look like in the XX female, XY male, and XO female who has Turner syndrome, and XXY, a male who has Klinefelter's, and XYY, a person with Jacob's syndrome, and then let's look at a person with XXX syndrome. So the person with XX female has one Barr body. A person who is a male, they have no Barr bodies. A person who has Turner syndrome, they have an X chromosome, no Y, but no Barr body. A male with Klinefelter's, XXY, they have one Barr body. So it doesn't depend on if the person's male or female, it depends on how many X chromosomes they are. Somebody with Jacob's syndrome, no Barr bodies. Somebody with XXX syndrome, they will have 2 Barr bodies. If somebody had 6 X chromosomes, they would have 5 Barr bodies. So if you wanted a calculation for the number of Barr bodies, you could say that the number of Barr bodies equals the number of X chromosomes minus 1. 2 X chromosomes minus 1, 1 Barr body. Over here, 3 X chromosomes minus 1, we have 2 Barr bodies. This one that we're subtracting, that's going to be the one that remains active. So these observations led the scientists of the day to propose that these cells, that human cells, and it turns out other placental mammal cells, have a mechanism to count the number of X chromosomes. They know that if there's 2, that one of them needs to be inactivated. How does it count them? How does it know there's 3 X chromosomes in this cell, 2 in this one, 2 in this one? How does it count? Okay, let's erase this and start thinking about how the cells are counting these X chromosomes. So let's draw a cell, and we're going to pretend this here is the nucleus. And we have 2 X chromosomes. So we're looking at an individual who is female. In the zygote stage, remember, both of these are expressed. The exact mechanism that the cells utilize to count the X chromosomes is not completely understood, which isn't too surprising, but we understand quite a bit about it. There's this region here called the X inactivation center. Or we'll just call it XIC. So there's an XIC over here as well. We know that it counts these somehow because if we were to take a cell, an XX cell, and mutate this X inactivation center, so there's a mutation on it, the cell will proceed to grow and not inactivate one of them. So we produce a mutation here, so the cell only now recognizes one X inactivation center. The cell somehow thinks that there's only one X chromosome. So now this cell will continue to grow and divide with 2 X chromosomes fully active. This will be lethal. So not only does that show that the cells are able to count in some way, but it's critical that it counts, because if it doesn't, you end up with 2 fully expressed X chromosomes and then death. So let's erase this down here. So let's draw this X activation center here again. It has 2 of them. Some ideas on how it knows to not express from one of them is that perhaps this one is shielded somehow. Maybe there's proteins that bind to it, so it doesn't express it. And it only does it to one of them. And so all the others get expressed. And saying expressed, let me tell you what's in this X inactivation center. there is this gene, and we mentioned it before, but we didn't say that much about it, called xist, which stands for X inactive specific transcript. You can just know it as xist. This is a gene in the XIC, the inactivation center. And what I want to do here is I'm just going to draw a little like shaded circle over this one, showing that somehow or another on this chromosome the xist gene is repressed. We're not going to turn on that gene here. As the embryo starts to grow and divide, and you have several cells, I'm going to erase zygote at this point, because now we're looking at a cell that's maybe 4 to 8 cells, or an embryo that's 4 to 8 cells. This is one of those cells from that's really small embryo. The xist gene now becomes expressed, and as it's being expressed, it's making an RNA. It's not a messenger RNA, it's a different kind of RNA that doesn't get turned into a protein. It remains an RNA, and it remains associated with this chromosome. Is draw a squiggly line around this X chromosome, and that's what RNA does. It wraps around the X chromosome that will become inactivated. Once this xist RNA coats the entire X chromosome, various proteins, and you can just know these as, so let's see, xist associated proteins. These bind to this xist RNA, and when they bind to it, they will begin to compact the X chromosome. So ultimately what you get is something that looks like such, where over here you still have your XIC, it still remains repressed, but now this X chromosome has become highly condensed because of these xist associated proteins wrapping it around it and condensing it. Now, previously when I talked about this Barr body, and this is what becomes the Barr body, I indicated that all the genes are turned off. But if you've been noticing, I've been saying a few things that are a little inconsistent with that idea. We talked about these pseudoautosomal genes, and when I mentioned these before, I said that these genes are on both X chromosomes, and they behave like autosome genes because they're both expressed. Well, if this is an inactive X chromosome, how can they both be expressed? Well, the reason for that is because it's only mostly inactivated. I know that's kind of lame to say that, but that's really what's happening here. This is very good at shutting down many of the genes, but not all of the genes. There's a few genes, not many, maybe somewhere less than 25%, that are still expressed at some level. So let's just put pseudoautosomal genes are still expressed. Does a pretty good job of shutting everything down, but for whatever reason, these genes we need to be fully expressed on both X chromosomes. And we know if they're not, that there are negative phenotypes associated with that. Now, as a question here, I'm going to ask, and maybe this will be part of the questions in class, is I said we don't really know how this is inactivated, but we have some hints and some ideas. And so what we've learned about other things in this semester, what do you think is going to inactivate this XIC region so that the XIST genes aren't expressed? Not only are they repressed in the early embryo, once this active chromosome is picked to be the active one, it's going to remain active for the rest of the life of this cell and all of its progeny cells within that organism. What would be an easy mechanism, so to speak, to make sure that this XIST gene remains permanently repressed? I'll put a question mark there. If I had some ominous music to play right now, I would, to set the mood of a mystery, but I don't. So what you'll have to do is just think about this for a moment and ask yourself, what is actually the mechanism that could keep that gene repressed? Alright, that's all I have to say about dosage compensation for this podcast. If you have any questions at all, please make sure you come see me and I'll help answer any questions you have. If not, I'll see you in class. Bye.